Hi, everyone. Welcome to this week's Ronin seminar. This week we've got Alex Lancaster talking to us about modeling cellular systems and apparently canalization. Take it away.
Okay. Hi. Thanks, John. Thanks to everybody. Welcome. And yeah, my name's Alex Lancaster. I'm a evolutionary biologist, and my background is sort of in the intersection of systems biology and evolutionary biology. And so today I'm going to talk about some of my research topics, just to give you an overview of some of the areas of things that I'm interested in. So this is probably not going to be a classic single topic research seminar. It's more of a sort of a taster. And also, I think for some of the people who know me through the Ronin Institute have probably not seen me talk about my sort of primary sort of research interests since I've been doing a lot of work in sort of helping build and talk about the Ronin Institute itself. So this will be a sort of, some of this will be hopefully new for folks. So, so one of the things I'm really interested in is this idea of stochasticity in biology. So Conrad Waddington, who was this geneticist from the 30s, had this really, what you might call very canonical picture of development, where he imagined this idea of this epigenetic landscape, where in development you would have, say, a landscape where if you started, at the top of this sort of hill with the rolling hills, and you drop marbles down there, they would, be depending on the exact direction of whatever the wind was blowing, that whichever direction, you would have some cases of development like on the left where you see the marbles go down one sort of rivulet, and on another case you have it on the other. So this sort of idea of epigenetic landscape became a sort of powerful metaphor for thinking about how phenotypes in biology can be influenced by small changes. So and you see this playing out in nature, where you have things like pattern formation in, say, fish, where you can get these really small changes in the nature of the patterns can generate lots of different widely different coat patterns, and so that's sort of the macroscopic level. The microscopic level, you have cellular noise, which is where you have genetically identical cells that have very different gene expression patterns. So this is a picture from an early 2000s paper by Michael Elowitz, where they showed that you could have a completely identical set of cells, and they would have a very different gene expression patterns. So shown by the different colors here, and this comes out because of basically the fundamental nature that nature is messy and noisy, and this has sort of all kinds of implications. And obviously in diseases as well, it's important to recognize that even in diseases like cancer, where you have genetically identical tumor cells, you can get, even in the same bolus of tumor cells, you can get subclones inside that tumor cells with very different phenotypic properties. So it's important to care about this, I think, for sort of two reasons. One, because it's really cool, it's really interesting. And as an evolutionary biologist, I'm kind of interested in how evolution can either harness it or filter it or do things with it. I sort of think of this as, in some sense, and this is maybe not a common way of thinking about it, but it's almost like, to my mind, it's like a population of cells can sort of explore phenotypic space in advance of genetic fixation. So it's kind of one means by which you can, noise can do diversification even before you have something like mutation to come along and basically fix it in place. And this is sort of the idea of, so in a way, in the cellular level, this is a sort of a molecular expression of what Waddington was proposing back in the 30s. The idea that that in single cell noise and genetically identical cells could be a molecular expression of this idea of this canalization, where you have a bunch of population of cells where noise changes the nature of the population, and it can allow a phenotype to arise that may be later fixed by a genetic mutation. The other reason is also there's fairly practical implications in terms of the mechanism of action of drugs and therapies, and I'll probably talk a bit about that at the end of the talk. So, so today what I want to do is I just want to give you 3 sort of vignettes of some of the research projects I've been involved in, just to give you an idea of the kinds of things that I'm interested in exploring and get some ideas from people in the audience. The first is I'm going to talk a little bit about yeast prions. I worked on yeast prions for about 5 or 6 years, still do occasionally. And they're a kind of a mechanism, what I think of as a sort of semi-stochastic switch. So it's a means by which a population can bet hedge. That is, it can explore a bunch of potential phenotypic alternatives that can be advantageous when environments are unpredictable. I'm going to talk a little bit about some work that I did for a long time ago on the evolutionary noisy transcriptional network. And lastly, I'm going to segue into some of the stuff I've been doing with my business partner, Gordon Webster, who's also, we together form Amber Biology, and we are now doing some work for commercial clients in modeling stochasticity in various systems, including the immune system, using a language called Kappa. Okay, so one of the things that's interesting about phenotypic switches is that, and noisy systems in general, is they can allow genetically identical cells to respond to stress in different ways. So for example, let's say we have a cell with 2 genotypes, let's say a white genotype and a red genotype. Now, if you put a population of those cells against a background, let's call this like light blue background, one way for the one means by which a population of cells can respond to a change of environment, say, for example, let's say the environment switches to a situation where the white phenotype is no longer advantageous. So that could be thought of as a form of stress. So let's imagine now you have the same population of cells are now against a background of a red background. Now, in this situation, the white cells are no longer adapted to the red background. So one means by which a cell population can respond is basically a sensor. And the sensor would basically somehow switch the phenotype to such a mode that the population is now compatible. So that's a good strategy when the environmental changes are predictable. But there is a cost to doing things that way, and there's a metabolic cost to maintaining that sensor. So depending on the nature of the rate of change in the environment, another means by which a population of genetically identical cells can respond to stress is via the stochastic bet hedging, which is what I mentioned earlier, whereby, let's say you might have occasionally a cell might switch to this alternative phenotype, the red phenotype, against a background of mostly white phenotypes. If it happens at a low rate, it's not so bad, because even though that individual cell might die, overall the population does not too bad. But if you take in the same situation where you have a stressful situation, then what can happen then is that it basically allows you to have a small fraction of the population can basically hang out when the environment changes and repopulate the population. So the situation, and other work has sort of established this, the situation of this, where this works, is when you have much less frequent but unpredictable changes. And in this case, there's no sensor required. So there's a lot of work that's been done in trying to explore the different, where the trade-offs happen and so on. What I was interested in is looking and applying this to the prions. Now, prions in yeast and prions in general are basically can be considered a kind of stochastic switch. So most people are probably familiar with prions from mad cow disease, but the basic it turns out that also prions exist in yeast and other kinds of cells, and I'll get to that in a bit. But the basic idea is that you have, if you have two proteins basically in a normal form, typically what happens when a prion forms is one of them will basically go rogue. So, and that means it will switch to an alternative conformation and basically switching shape. And when a regular copy of the same protein encounters that switch that counts as the new shape, what it will do is it will actually sort of recruit it together, if you like. It will basically add it into this aggregate. So they basically become like these sticky pieces that sort of basically stick together. And so now you have one of these prion particles that can extend by recruiting new copies of itself. I think of this in a way, it's almost like zombification here. You know, you have your prion population, you have your uninfected zombies, and they get progressively recruited as the prion process goes on. Now, this would be interesting if it just happened in a cell, but it turns out, just in a single cell, it turns out that because these prion particles, once they get big enough, they can actually be cleaved into little pieces, and those new particles can then, initiate the next round of prionization. And key for our but what we're talking about here is that some of these particles can be inherited cytoplasmically. So this gives a way for cellular state to be inherited across generations. So once it's in what they call the prion form, that's where it all goes to capital letters like that, especially at least in yeast, what can happen is the prions can actually be inherited by daughter cells. So this is a self-perpetuating prion state that is heritable. So as I said before, you mostly think about these in terms of mammalian prions. We all know about mad cow disease, but it turns out that at least in some cases in yeast, prions can actually be beneficial. So this is a growth curve showing that a wild grape must containing prion can actually increase its optical density, meaning increase the density of population. However, when you remove the prions are no longer that beneficial. So, and they've done these experiments across lots of different conditions to show that certainly in some fraction of cases, there's definitely there's an advantage to having prions around. So how does this work? Like, what's going on under the hood? How do these prions? Why would they even be advantageous in any sense at all? So it turns out what prions do in the cell, and this is true at least in this case of the most commonly studied yeast prion called SUP35, is that what they normally are doing is they're hanging around in solution in the cell, and they will participate in what's called the translation termination complex. So that's basically the thing that sits very close to the end of the stop codon on a messenger RNA. So here's your messenger RNA here. Let's say you have a bunch of you have a ribosome that's trying to basically translate this RNA into protein. What it typically does is it trundles along this mRNA, and it's extruding these polypeptide chains out the back of it as it's basically reading the genetic code. When it gets to the translation termination stop codon, it goes, haha, I'm done. Okay, I can detach. Ribosome is tagged, and you're good. The reason this functions normally in the normal case is that it basically the SUP35 is basically doing its job by participating in the termination complex. When you have a prionization of the SUP35 complex, what happens is that it forms these aggregates. It basically no longer can do its job anymore. So the termination efficiency goes down. And what that means is that basically the ribosome has at some probability a chance of basically continuing on, basically ignoring the stop sign, driving to the intersection and adding up a few polypeptide chains at the end. So what that's going to do is going to basically create a bunch of tails at the end of your proteins. So you're going to get an alternative trait. The cool thing about this is just temporary. So aggregates can be removed, translation termination can be restored, and we're all back to where we started. So then the question becomes like, why would you use a prion and not a mutation to do the same thing? Well, you think of this from a population point of view. Let's say we have a prion that switches at some rate, let's call it, basically does exactly what the prion does, but it does it by mutation. If you're in the population that disfavors the prion phenotype, those are going to be eliminated quickly. But in situations when you move to a stressful environment where the mutant might become advantageous, your mutant is going to basically survive, whereas your non-prion versions will, or rather non-mutant versions won't, and they will increase in frequency, take over the population. But you got a problem when things switch back. Because now you're stuck. There's no way to get back to where you were, and that subpopulation at least could go extinct. So contrast that with the prion situation where you have the same thing. You might have at some rate you have a appearance of a prion. Again, that will rise to frequency in the population. But when you switch back, you now at some probability have the ability to lose that prion phenotype, and that subpopulation can survive. So to sort of ask the question, like, well, under what circumstances, in terms of evolutionary, in terms of the major evolutionary forces, the question then becomes like, what are the sort of parameters under which a system like this can actually work? Like, what are the population sizes? What kinds of gene flow? If we have let's imagine we take the individual subpopulations and put them in what's called a metapopulation, where now you allow basically migration between these what I'm calling demes in the subpopulation. What happens when we have that? Can this actually be selected for? So what we did is we created this population genetic model where we had the demes, the basically subpopulations, all exchanging individuals. We parameterized it by things like the mutation rate and the rate of prion appearance. And what that allowed us to do, and there's a whole bunch of math, and I'm not going to go into, but basically under the hood, you have a stochastic model in each deme that's embedded in a deterministic model that determines which at the metapopulation level. So that's basically controlled by a differential equation. So at the end of the day, when you sort of put all the parameters in, you turn the handle, and you basically create these like phase spaces where you vary. In this case, I'm varying the rate of prion appearance, the mutation rate. What you can do is you can generate like a three-dimensional surface where you have, and then based on it's like a contour map, and here I'm just coloring like a heat map showing you where the parameter ranges are such that the rate of prion appearance. What it basically showing is what the rate of prion appearance and mutation rates are that will favor the prion system evolving. So that's all nice as a model. What does the experimental data say? So a few years ago, I was working with some folks in the Lindquist lab. Susan Lindquist was a biologist at MIT. She's since passed away, very sadly. And she's been working on trying to understand prions for decades. They did some stuff we did with some of the folks there was that we basically were able to take my model and then compare it with some of the data that they collected in the lab, where they looked at the the rate of appearance rate using something called the fluctuation test. So here we're basically basically able to place on this landscape for different kinds of prions. One is the classic prion, which is the psi one, which is the sub 35 that's in green, but also a number of other different kinds of prions here. So at least there's some, the jury's still out on this stuff. It's still quite controversial, but at least there seems to be some evidence that suggests that prions can be, at least have the potential to be selected. There are people in the field who really don't like this idea, and they maintain that prions are disadvantageous even in yeast, and they won't entertain any other idea. But to my mind, at least there's some interesting phenomena going on that we still don't fully understand. So the takeaways from this for me are that there's at least some evidence that prions may be selected for their bed hedging properties. And also it turns out that the prions themselves, their classification is actually been expanding the last couple of years. So it turns out that there's a lot of other, when you start looking at what's going on in the cell, there's a lot of other different kinds of proteins that have prion-like properties, and some have been established as bona fide prions. One in particular is called MOT3. It's actually a transcription factor. There's also this global transcriptional regulator. So we're probably at the beginning of trying to figure out what's really going on in prions. And it may turn out that what we think of as a classic prion is just a special case of some more general behavior in the cell where proteins are constantly shifting their protein conformation in response to the environment. So it's kind of the beginning for all of that stuff. I think it's also an interesting model for other stochastic bed hedging processes in cells. So, for example, I mentioned earlier, cancer tumors seem to exhibit some behaviors that can be consistent with the idea of being a bed hedging strategy. So, if you're a cancer, you might want to evade some sort of any kind of therapy. It'd be handy if you could hide out in an alternative phenotype and let your brethren basically take the hit. And then once they're all cleared out, you go in and repopulate. So, and this is actually found in the bacterial persister cells, which is actually probably the most well-studied bed hedging process, whereby a population of cells, a tiny fraction of bacteria will switch to an alternative phenotypic state and ride out the application of antibiotic resistance, and then repopulate. That's another major reason why you should always finish your antibiotic courses. So yeah, my feeling about this is the mechanisms are going to be very different, but they all face similar evolutionary pressures. So, and I think like stochastic modeling of systems like this hopefully may help design different therapeutic strategies, and I'm going to talk about that more towards the end of the talk as well. Okay, now I'm going to switch gears quickly to talk about briefly about another project that I was involved in, where we were looking at now switching to moving from translation into transcription, and one of the things that's really interesting about transcription is that it's rampant, and that's because these transcription factors, which are, for those who are not biologists, are basically these proteins that bind to the DNA and switch genes on and off. So, and they often are in very small number of copies. So you may have like 1 to 5 to maybe 10. And so, in the cell, it's a noisy place. So it's sometimes amazing that transcription, even genes, ever get switched on at all. So one of the questions I'm interested in is sort of what's the role of transcriptional noise in shaping evolution of these transcription networks? And in particular, there's been a lot of work done in looking at what might be called network motifs. These are a sort of topological mini structures that are within the transcriptional networks that can be analyzed from sort of large scale omics and big data approaches to systems biology, where you look at like structure of these sort of mini patterns. And it turns out that some of these patterns seem to be overrepresented, and they can almost function like little circuits. And one of the hypotheses is that that people have been putting forward for these particular structures that have been selected for particular functions. In particular, one has been proposed for filtering noise. But the question is, have they been selected for this function? So these are the kinds of questions that stochastic modeling and system biology, coupled with an evolutionary approach, I think can help tease out. So we did this kind of crazy, this is work I did with Joanna Maisel at University of Arizona. And we created this sort of platform, if you like, where the idea was to model, basically build fairly realistic models of transcription where the parameters were drawn from yeast data, but they were small enough to be tractable, meaning that they weren't a kitchen sink model where you had every possible interaction built in, but they were realistic enough to be representative, but they were small enough to be tractable to in silico evolution in a reasonable time frame. And the idea is that we build models that are, this is what you might call an ensemble approach, where you are interested in the typical properties of networks, rather than building a specific network for a specific sort of like known thing, a known pattern. A known network, but the parameters themselves are drawn from data. And again, this is what I think of as an agent-based modeling approach. So here you're representing the individuals in the cell in some sense, and the important part of this is it's a good way to capture the true heterogeneity and get full transparency on what's going on in a model. And this is, I think, something I'm sort of building a little bit on Gordon's seminar from a couple of weeks back, if people were have had to happen to be here on that, where agent-based modeling or rule-based modeling is a really important way to be able to get at that. Okay, so here's the model that we built. This is just a cartoon model that represents just 3 transcription factors and 2 genes. The model itself, when it was running, could have anywhere from 3 to about 19 genes. But at the simplest, we imagine that, let's say we have a signal coming into a cell. It can bind to the transcription factor site, and the transcription factor sites are just upstream of the actual gene itself, shown here in this little box with this arrow. This is a classic picture of a gene transcription for non-biologists. And what that will do is that when you have transcription, obviously, you get your messenger RNA. Messenger RNA can be turned to protein, and so on. And the key here is this protein can not only feed back to, can not only also bind to another transcription factor further upstream, another site upstream on what we might call an effector gene. It can also feed back to the gene you just produced. And so basically, once we put all of these different features into the cell, so we have, we represent basically the feedback loops, we represent all of the transcription factor binding sites, and we also model things like protein decay and RNA decay, all that good stuff. The key thing here is that what this does is that it allows you to wire up a bunch of genes and proteins endogenously, meaning that the structure of the DNA binding sites themselves create the interaction network, rather than being a arbitrary rewiring of sort of nodes that are black boxes. And to make things really complicated, we also added inside this process, we actually modeled the 3-state transcription initiation. So we actually kind of exploded what's going on in here and modeled like some of the processes that were going on, say how you had repressor. You know, I won't even go into all this stuff. It gets too complicated. But basically, there's a bunch of stuff that's going on underneath the hood as well that can includes all. This is all the sort of noisy. This is also where a source of stochasticity comes from. So the idea is that once you have a bunch of these transcription factor binding sites, you can and each regulatory region where the transcription factor binding sites were initiated by a random stretch of 150 base pairs, and then you would scan the base pairs based on the sequence of the transcription factor binding sites and basically identify all these sites that had to be wired up. And what that gives you is a way not only to wire the networks up so that you know which genes turn on which other genes, but it also generates these maps of transcriptional logic. So here I'm showing you on the left, you have an adjacent but non-overlapping transcription factor binding sites. You can actually interpret that as an AND gate. And what I mean by that is that if you have a signal coming into your cell, in an AND gate, you need to have both the signal and something else to affect the actuator. So what that means is that you can actually switch the gene on versus a OR gate, which means you only need two. So what you can do is you can basically scan the patterns of transcription factor binding sites and identify all the different potential gates that are the potential motifs based on these patterns that are in the transcription factor binding sites, based on at whatever level they happen to be at now. Okay, so that's the sort of functional biology part of it. So then we did something even crazier, which is basically, let's imagine we start with a genotype, and that's just a DNA sequence. We can use that genotype to build the network of interacting transcription factors in the cell. What that gives us is a phenotype, which is basically the output of the transcription factor concentrations over time. And we can use that phenotype to compute a fitness. And the fitness in this case was measured as the output of the affected gene as a function of whether it was advantageous in a particular environment. I won't go into the details here, but sufficient to say that there was one of the things that affects the fitness is the cost of gene expression. So we say that, you know, producing a lot of stuff, you know, is a costly thing, but and then there are a bunch of other things that go into that. But at the end of the day, what you get is instantaneous fitness for that particular network. And from that, we can then define what that means in terms of an environment, whether a signal is advantageous or disadvantageous. We can basically mess with the what we want to evolve in the cell. So once we do that, we can define for any set of phenotypes whether it's close to fit phenotype, and we do mutation and selection. So what we do there is we generate multiple mutations, and the mutations take the place of things like switching or changing the DNA sequence in the cell. There's also an option for gene duplication. There's a bunch of things that can go on. And you rerun the cell and you see whether or not you have a fitter network. And you keep doing that until you have a new genotype that has a higher fitness than the resident genotype. And this is sometimes called origin fixation model in population genetics terms. But it's not important to know too much about that, other than you can basically evolve the phenotype of interest. And then, of course, then you get a new genotype, and the evolution continues. So we have a preprint out on this, and this is all work that was done by Kung Zion, who is a graduate student at Arizona, and he was able to find that coherent type 1 feedforward loops were able to filter out noise, which was really interesting, because this is something that's been proposed in literature, that these feedforward loops evolved for this purpose. And one of the reasons that this has been proposed is that when they do, is basically when you look at the network data and you do omics data, it looks like the these motifs are overrepresented. So there's sort of like a tendency to do a sort of evolutionary just so story where you say, oh, well, look, it's overrepresented in the data, so it's obviously going to be under selection. And so one of the things that we didn't think was sufficient for this is it's not a really good evolutionary argument. You can't just say, oh, well, we see it overrepresented, therefore it must be selected. So one of the cool things that this model is able to do is basically test that idea. So what we're able to do is we're able to show that these kind of feed-forward loops actually do evolve under selection for filtering out noise, but not under control conditions. So you can basically set up situations where the filtering is basically neutral, or it has a deleterious effect, and we're able to show that under control conditions, the frequency of these feed-forward loops appear much less frequently. So there does seem to be evidence. That evidence that these feed-forward loops actually do evolve for the purpose that's been given before. But until now, there wasn't really a good evolutionary model or argument for that. And the other cool thing that he found was that a lot of focus was on these feed-forward loops, but you can have other topologies. So there was another interesting topology called an isolated diamond, whereby you would the effector protein, the output protein, was basically integrating signals from two pathways rather than just the one in the feed-forward loop. And it also evolved to filter out these transient signals. But unlike the other feed-forward loops, it was also effective at filtering what you might call intrinsic noise. That's the noise that's due to stochastic effects in the binding itself. So anyway, there's a lot more to this project than I can possibly give here, but you can go and check it out there. There's the preprint. So anyway, so that's sort of like it's kind of the end of some of the evolutionary stuff that I've worked on. I'm still very interested, and I'll summarize at the end. Now I want to kind of segue a little bit into something a little bit more applied, which is some of the agent modeling work we've done as therapeutic intervention. And I think this is, again, it's important for all the reasons that we've talked about in evolution, but it's also important for practical reasons, because you really do need to take into account the stochastic nature of the systems. When we're injecting molecules into tissues of whatever type we're interested in, you really want to know what those perturbations are doing in a very specific way. And as we've shown before, as I've kind of like shown so far, is that small variations in copy number can make a big difference to cell state. So this is just a picture of some stochastic modeling that was done by this group here, and they were interested in looking at the phase portraits. So that's basically looking at when you vary the fraction of these different regulators, and there's about 3 different regulators they're varying here. I think there's cyclic, and this is all part of, I think this is called cyclic something. I forget exactly what this is called. Anyway, you have, these are basically 3 important kind of sort of knobs, if you like, on that are important in regulating the cell cycle. And as you can see, this is sort of the sort of global picture, and you can see like, you can imagine if you're up here in one location on your phase space, that you get a small variation, you can be in a whole different location. So this is work I've done with through Amber Biology. Folks might know Gordon from our Ronin seminar before. And one of the things we've learned from, and I've learned, and Gordon and I have both learned from our earlier modeling efforts, including all the stuff we did before, is that stochastic modeling is hard. And it's easy to make mistakes. It's a lot harder. It's a lot harder in some ways than just regular ODEs. However, ODE models have many drawbacks as well. And I'm not trying to say one is better than the other. They all have their place. I use a combination of both. But but it has been, in terms of like how to do stochastic modeling, it has been a lesson in what I call unlearning. When I was actually out at Sydney for the early part of the year, they had these pictures all over campus. They just, someone in their infinite wisdom of campus had decided that to plaster these things about unlearning all over campus, which everyone thought was quite hilarious. So I'm not sure. Probably someone in marketing got a big raise for this, but I don't think it had the intended purpose. Anyway, so yes, so back to yeah. Maybe I'm at 12:45 now. Maybe before I launch into this next section, I'll just pause for a second. Does anyone have any questions so far, or are we happy to wait till the end? I feel like I've been talking nonstop for about 40 minutes here. Hello.
I have to run to work, but I just wanted to say this was fabulous. I've learned a ton.
Okay, who is that?
Skylar.
Oh, Skylar. Hey, Skylar. Okay.
Hey, nice seeing you.
Yeah. Yeah. So I'll continue, and yeah, and feel free to. It's for some reason I don't sometimes see the hand waving here. So, but do feel free to write something in the chat as well. Okay. So yes, and this will be familiar for folks who were at Gordon's seminar a few weeks ago. One of the big drawbacks with ODE models, as he said before, was that you get this combinatorial explosion of species. So just a very simple model where you have a kinase and a substrate, and it can bind at 2 different sites on a substrate. You still get 13 different molecular species and 13 rate equations. This is if you're doing it using ODEs. Now, if you want to just add a very simple change when you add a complementary phosphate to the model, you have to then rewrite all the original rate equations, and you have to add another 21 new ones. So this is going to be a problem when you're building even more complicated models than the models that I was sort of talking about earlier, the more evolutionary models, which are a little more on the stylized end of things. So just for the, say, EGFR signaling system, you would need to write about 10 to the 9 different possible, 10 to the 9 different equations. So that it's clearly not feasible to do that when you want to model really complicated networks. So Kappa is a language that can at least help alleviate some of these problems. And the idea of Kappa is it's a language is much closer to molecular biology than differential equations are. And it's all open source and available there. You can download it and play around with it. There's a web version of it too, which is pretty cool. And the key thing with Kappa is there's 5 different rule types. So you have synthesis, which is just obviously the production of a molecule. And here I'm sort of showing you the cartoon picture at the top. And then underneath it's the Kappa syntax that represents that transition. You have degradation. That's where you have obviously the molecule disappears. Again, this is what it looks like in Kappa. Binding, unbinding, and there's the syntax gets a little more complicated, but hopefully you can sort of see that there's a relatively straightforward way to represent these processes. So one of the things we've done for some of our commercial clients is build models of sepsis. So that's an immune system response triggered by infection. And the focus of this was we were interested in determining levels of 2 anaphylotoxins in the immune system pathway. And the goal of the model is to try and figure out how, like, changing, modulating these toxins by just small molecules can affect that pathway. So this is like trying to figure out, like, how, you know, and again, like, there's some, you know, cases when some of these are in small copy number. How are these, like, how is this noise? How are these stochastic processes affecting the overall outcome, therapeutic outcome? And this is part of, I think, an important process in really coupling some of these computer modeling things that we're doing with more closely with lab experiments. So just to give you an idea of what these, and apologies for people who've seen this already from a couple of weeks ago, but this is the same, more or less the same slide. This is a stochastic rule-based model built using Kappa, and here on the bottom you can see what the molecule would look like in the cell, or so stick model, and when you have two agents that interact like this, so this is one agent here on the left, and the other one is on the right here in the orange. What Kappa does is basically when you have agents of a certain type, when they're around, it will look through its list of rules and say, aha, this rule is the one to be applied here. And what this rule is showing is that you can have binding between the C3 site and the R3 site. So, and it's also the fact that it goes backwards means that this rule is also reversible. So this is a way, so if you were scouring through literature and you found out that this is, this more or less represents some sort of signaling pathway, you can encode this into Kappa. And then from Kappa, once you've created a bunch of rules, so you write these out just in a text file or however you want, it can create these, do things like create contact maps. So that's like a wiring diagram, kind of going back to what we were talking about earlier with the transcription factor network. If I was to build that model again, I'd probably use something like Kappa. And this is sort of something that's, kind of close to what molecular biologists do in the back of an envelope, or at least it's closer to that. So that's another important factor here, is that getting people to use this or being able to explain across disciplines, it's important to try and use the idioms of people from those fields. And Kappa at least gets you a lot closer to that. The other cool thing is you can do virtual experiments. So for example, we were able to do an experiment where we basically could perturb the system. So we took a factor Q, which I can't say exactly what it was, and took it down 20% of the level and compared the outputs. And so we could, then basically ask questions like, this could be like a therapeutic intervention. We stick something in the cell, look at what the output equilibrium ratio is in the normal case, and then sort of basically run the in silico experiment. So we're in the process of doing all that kind of stuff. And Kappa has also been used to model a lot of different pathways. So it's been used to model pathways of insulin, dopamine, the Wnt signaling pathway. So it's a very versatile set of tools, and that's still fairly underused at this point, I would say. It's still hard to get biologists to use this stuff, as I think Gordon said in the previous. There's still a bit of a gap there, but we're working to fix that. Okay, so given this an academic talk, I sort of have my usual sort of future direction slide here. I'm still interested in exploring some of these evolutionary questions. Picking up on some of the threads of the earlier part of the talk. I think one thing that'd be kind of interesting and cool would be to combine some of the transcription factor networks, but also examine how evolution might be affected by transcriptions that are also prion-like, meaning that they could also switch. So some questions there might be like looking at the optimal number of switches. Is the cell exploiting them? I would probably use something similar to the whole cell transcription network approach, but maybe you model it using Kappa. These are all sort of things that I'm thinking about now. And also potentially applying these things to other systems as well. From the systems biology therapeutic application point of view, obviously there's a lot of scope to expand to other pathways and systems. We're working with other collaborations, industry partnerships to try and get more uptake of these kinds of tools and explore ways in which we can either expand the tools or continue to build models for clients and just basically help build the community around a lot of these agent or rule-based modeling tools. And I think the time is right for a lot of this work, because for a long time, I think system biology oversold itself, and I think it went into hibernation for a while, but I think it's about ready to come back. So yeah, with that, I'd like to have my acknowledgement slide, and thanks to all the people I work with over the last, like, I guess 10 years now. And obviously thanks to John and for the Ronin, and to Gordon, my Amber partner. And you can find my Ronin page here, and contact us through Amber Biology. I have a Twitter feed and all that good stuff. So that's, I think, my last slide. So yeah, that was a bit of a whirlwind overview. Thank you for your attention.